Hey folks, and welcome to another edition of Zog Science. Today we're going to be continuing our discussion of biotechnology, and we're going to get into genetic testing. So one of the main techniques that we're going to be talking about is gel electrophoresis. And gel electrophoresis, as the name might imply, is going to use electricity to push DNA through kind of a maze. And that maze is generally going to be made of agarose gel, which is a sugar. It's not always going to be made of agarose. There are different applications, and there are different consistencies of agarose that are going to be used. But in general, it will be agarose. And what happens is that as we push the DNA through the maze, larger pieces of DNA are going to get stuck and are going to move more slowly than smaller pieces. So what that means is that our longer pieces of DNA are going to be more towards the beginning of the gel, whereas the smaller pieces of DNA are going to have moved down the gel. And what that does is it allows us to separate fragments of DNA based on their size. So why does that work? Well, DNA is negatively charged, and it's negatively charged because of the phosphate groups that are all along the phosphate sugar backbone, right? So what happens is that we load the DNA on the negative end of a chamber inside of an agarose gel, and then we run a current through a buffer. That buffer a lot of times is going to be TBE, which is tris borate EDTA, and that buffer allows electricity to pass through. So we have to have some sort of medium for the electricity to pass through. And what happens is that the longer molecules aren't able to move as far. The shorter molecules are able to move much farther. And this is going to create a pattern of DNA based on the size of the fragments. Now, how do we actually get these different size fragments? Well, as we learned about last time, we have restriction enzymes. So what we do is we digest the DNA. We mix it with with enzymes in order to break it down, and it's going to cut it in specific sequences. And this is going to create a pattern that is going to be specific to whatever it is that we are looking at. So what this does, again, is it creates what we call a DNA fingerprint. And everybody has different DNA. We'll talk about why a little later. And that's, again, because the DNA is cut with restriction enzymes, and it's going to produce a unique pattern. Now, coupled with gel electrophoresis is PCR, and the guy who invented it got the 1993 Nobel Prize, although he came up with it in the 70s. And it's a method for amplifying specific sequences of DNA. In order to do DNA polymerase chain reactions, you have to have polymerase molecules. Specifically, we use Taq polymerase. That Taq polymerase was isolated from thermophilic bacteria that they found in hot springs because it functions at higher temperatures. We need polymers. Specifically, we need the nucleotides for the DNA. Then we've got our primers, which are RNA primers, and we need the starting DNA sample. It doesn't have to be very much. We just need some of it. So here's the steps. We've got our target sequence of DNA. What we're going to have done is we're going to have created some primers that are going to bind to the ends of that target sequence. All right. So we've got 3 steps in each cycle. The first step is denaturation. So we increase the temperature. That is going to separate the DNA, 2 DNA strands, because they're moving too much. They have too much kinetic energy. They get pushed apart. The primers will then come in, and they will attach at the sites. And you can see that happening right here. And then we've got our Taq polymerase that's going to come along, and it's going to add new nucleotides. So we now have a new sequence and 2 new strands of DNA. The cycle 2 occurs, and now we've got 4. Cycle 3 occurs, and we now have 8. But you notice that some of these molecules are beginning to be just of the target sequence. And that's because the DNA polymerase can only add on to the free 3' end. And so we end up with, eventually, after many cycles of this, of our DNA ending up as almost exclusively just the target sequence that we want to have. So, as per usual, I've got a nice little video that shows us this in a little bit more detail so we can kind of get an idea of what this looks like.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
All right, so I hope you were able to kind of see how, as that video kind of progressed, we're able to get more and more of just our target sequences that is produced. All right, now, what we really do is we really use both PCR and electrophoresis together. We use them side by side. We use them in some pretty cool applications such as forensics, genetic testing, and also health. So in forensics, the suspect's DNA can be compared to the DNA found at a crime scene. So what happens is that we take a little piece of DNA, and in CSI and other crime shows, they talk about, oh, well, we just got this little piece of hair that has a little bit of skin on it. And what they do is they use PCR to amplify that DNA to a point where you can then run it through a gel. Because you need enough DNA to put through a gel in gel electrophoresis to where you're actually going to get some bands to be able to see it. And so then we put it in with some restriction enzymes. Those are going to cut the DNA. Remember, everybody's DNA is different. And so therefore, when they get cut, they're going to be cut at different places, and we get different bands that are produced for every person. And then we can analyze our results and see which DNA is the same. We can do something very similar with a paternity or maternity test. So the child's DNA is going to be compared with the potential parent's DNA. And the child's DNA fingerprint is going to be a combination of both parents, right? So if we kind of look at the example over here, this offspring has to get every single one of those bands from one of its parents, right? It's either going to get one of the bands from the mother or one of the bands from the father. So you can very easily eliminate people who are potential fathers based on whether or not they are contributing those specific bands. Another application that we have is in health. So different alleles are going to produce different bands, right? So if you have a mutant allele, it is going to produce a different band than a normal allele would. So what we can do, in this case, we're looking at hemoglobin, right? Or rather, I guess more specifically, beta globin. And the sickle cell allele, because of the mutation, it actually disrupts one of the restriction fragment sites, and therefore we get a larger band fragment that is produced instead of 2 smaller ones. Therefore, we get different bands that show up when we look at a gel electrophoresis. So what they do in a lot of health things is that they will compare people to one another, and you can run a DNA gel electrophoresis on somebody and see whether or not they have a specific allele. Therefore, you can know whether or not they have that disease. So this is kind of a critical part of this. Why is everybody's DNA different? Because this is kind of the basis for a lot of these things. Well, we have RFLPs, or RFLPs, and that is a restriction fragment length polymorphism. And those are basically differences that are going to change the length of a restriction fragment. And so they're obviously, therefore, they have to either make a restriction fragment significantly longer or shorter, or they're going to have to disrupt a restriction site. So we have VNTRs and STRs. Those are variable nucleotide tandem repeats or short tandem nucleotide repeats. And basically, those are going to make the restriction fragment lengths longer or shorter because they are repeats of a specific DNA sequence. So VNTRs are generally going to be 5 or more nucleotides that are repeating. STRs are generally going to be 2 to 4 nucleotides that are repeating. And so basically what happens is that some people have an allele that has, or I guess not necessarily an allele, but they have a chromosome that has more repeats or less repeats of either of those, and therefore they're going to have different fragments that are produced. In general, these are used in identity, although they are sometimes used in health as well. SNPs are single nucleotide polymorphisms. So these are going to be places like in sickle cell where we have a specific nucleotide that is changed, right? That is a point mutation. That can lead to a single nucleotide polymorphism because we have a single nucleotide that is affecting a restriction site. Therefore, different length fragments are produced. And again, the only reason why we care about the fact that they produce these different length fragments is that when we run them through a gel electrophoresis, we're going to get different bands and therefore a different pattern that is going to be produced so we can compare people to one another. Something I've mentioned in class, but I just want to make sure I mention it here, is that we don't just look at one of these. We look at many of them, particularly when we talk about identity, because people can share some. However, if you look at enough sites, you will most likely be able to see some differences. So I think that's all we've got to talk about today. Next time, we'll talk a little bit about cloning. So be prepared for that. Thanks for watching, and I'll see you next time.